Thank you, Pat. And now to our symposium. Our first speaker, Dr. David Botstein, received the Gruber Genetics Prize himself in 2003 and has since served on the advisory board that selects annual recipients. He is a renowned geneticist and educator and director of the Lewis-Sigler Institute for Integrative Genomics at Princeton University. His fundamental contributions to modern genetics include the development of genetic methods for understanding biological functions and the discovery of the functions of many yeast and bacterial genes. In 1980, Dr. Botstein and 3 colleagues proposed a method for mapping human genes that laid the groundwork for the Human Genome Project. In the 1990s, Dr. Botstein collaborated with P.O. Brown in exploiting DNA microarrays to study genome-wide gene expression patterns in yeast and in human cancers. A graduate of Harvard, he earned his doctorate from the University of Michigan. Dr. Botstein taught at MIT and Stanford University School of Medicine and was vice president for science at Genentech prior to joining the Princeton faculty in 2003. Please welcome Dr. David Botstein.
Well, thank you very much for the nice introduction. And again, it's a pleasure to be here and see the Grubers and to talk to you about genetics, which I'm very into, as you'll see. So you may all recall, I'm assuming this is quite a mixed audience, you may all recall that when the idea came up that we should sequence the human genome, there was a great deal of controversy. And there were very many people, some of them even here at Rockefeller, who were skeptical that this actually would produce any interesting science, let alone produce anything interesting for society. And so about a year ago, I thought that it would be good to put together a sort of summary of well, you know, what have actually been the deliverables from this activity. And that's what I'm going to do. Now, I'm going to do it in a sort of egocentric way. I know my own work best. We've exploited the genome sequences in various ways, but I'm by no means going to talk only about our own work, but the examples will be local, as it were. But I hope you get the idea. This is sort of a tryout of this thing. Okay. So the yeast, after the controversy, the yeast, worm, fly, plant, and human genomes were sequenced, and some attempt was made to find all the genes, although this has been an ongoing saga. The number has been going up and down. It's not clear that all the important genes make proteins. It's not clear how many of the RNA genes are how important, and there's still a lot of DNA. It all used to be called junk that wasn't worth sequencing, and more of it is becoming important, and I don't think that's over yet. But I've cataloged for you the protein encoding genes, and one of the really interesting and in some ways dismaying to some ideas is that actually there are almost as many genes in this very simple unicellular organism as there are in worm and fly and plants, and they have about as many genes as we do. And somehow this doesn't sit well with our sort of species elitism, I guess. However, all of this activity did, and I'm going to perseverate on this theme for a while, did produce what I regard as the number one biological thing that came out of the genome, and that is that the basic cellular functions, the things that make one cell into 2 cells and that keep order and the housekeeping, if you like, from metabolism to cell division and cell surface and cell signaling and all that sort of stuff, all of those basic cellular functions are carried out by genes that are essentially the same. That means that you can recognize who was derived from who for each gene, and not only that, but you can usually assume that if you really got the function right in one of these organisms, that in another organism it'll be really similar, at least at the nuts and bolts level. And so this is the sort of, I like to call it, I've got a lot of physicists I deal with, so I have a grandiose way of saying this. This is the grand unification of biology. Most of my life I suffered from people telling me whatever I learned was useless for some other organism. And of course, we have the last laugh, so we enjoy that every now and again. And in fact, the whole problem of associating biological function with a particular gene or a particular sequence has become the preoccupation of a very large fraction of the biological community, for good reason, because all of this ability to transfer understanding from one organism to another has made it really worthwhile to try to find out what a particular class of gene does in any organism, because then it gives you a big leg up. And very often it's easy to do an experiment in yeast or flies, and in fact the business about the clock that the Gruber Prize was given for neuroscience this year really was worked out, I hate to tell you this, in flies, but it all applies to humans too. Okay. And just as an illustration of this, you can see that if you just take the sequence of amino acids of a protein and you look at the similarity, you get all kinds of ranges of similarity, but some of these are very high. And what is interesting about this is not so much that these are all very important genes. Like, for example, I take an inhibitor of this every morning to keep my cholesterol down. Yeast don't even have cholesterol. They have something like cholesterol, but not cholesterol itself. The tubulins separate the proteins and so on. What's interesting about this is that you can knock out a gene in a yeast. There's no special paperwork required to do that experiment. And you can knock out a gene in yeast and then put the sequence from some other organism, let's say the human or the cow or the mouse, and ask the question, whatever consequence there was from the loss of the gene, can I recover the function from the gene from human? And the answer most of the time for this class of highly conserved genes is yes. So think about that. It means that if I have a Model T Ford outside and a Mercedes, and the Ford won't run, what is the probability that I can reach into the Mercedes, grab a part, put it in the Ford, and drive away? And the evolution of automobile engines is 100 years, plus or minus, okay? This is billions, 3 billion years or so between the protagonists here. Okay, so that's the top line, most important result that we got. So I take the fruits of the genome more generally is the quantitative understanding of evolution by looking at sequence, the grand unification of biology, the uses of variations in sequence within a single organism, and then the business of being able, through the gene, to find out what might be wrong in an inherited disease, and then DNA diagnostics. And I'm going to say a little bit about all of these. But those are all real here today results of the genomes. So I'm going to start with Darwin. This is, the year of Darwin again. There's always a year of Darwin. Darwin went, Darwin came, and whenever we have an anniversary. But in the most recent anniversary, the Museum of Natural History had a big exhibit, and I saw this diagram, and I was blown away, because Darwin had no way to actually make this diagram in the sense of the way we make them now. But this was his intuitive insight. This is what it really looks like if you do it with ribosomal DNA rigorously. Looks actually Darwin wasn't far off. A better way to look at it is this way, because you can read everything. And I just put this up partly to impress you with the fact that we're all start from a common ancestor, bacteria, and the new class of bacteria, and us. And also to point out that us is a very small corner of this universe. Okay? Yeast is just next door to the animals. The two I was comparing, but in fact the principle goes way back, and I'm going to talk about that. So how do we do this? Well, the way we do this is with computers. It's very important to understand that had the rate at which computers become productive not been as fast as it was, we would not be here with this whole DNA sequence stuff. Because what we can do now on a machine like this in a couple of seconds is we can look through all the sequences that have ever been determined, and with a little snippet of sequence that we have just found, and ask who's most similar in the whole world. And this was done for a particular bacterial gene called MUT-S. It's a DNA repair gene. And what you find is that the closest similarities, all the black is identical. Closest similarities are from yeast and mouse and human and so forth and so on. It's not one yeast and another yeast and another yeast. It's yeast, mouse, human. Think about that. After all these years, these are still the most similar. And if you look even among the bacteria, you see evolution in action. That is to say, the common ancestor began with one copy of this gene, MUT-S, this gene MUT-S, and then it duplicated, and some of today's bacteria have lost one copy, and some have lost the other copy, but most of them have now two copies. And why would they have two copies? They would have two copies because the two copies have learned to do different things. If you want one gene to turn into another, one function to turn into another, and the first function is essential, you can't just tinker with it while it's running. You have to make another copy and then tinker with that. And that's the fundamental principle, and you can see that principle just from analyzing sequences, and all the time. And in fact, by the time the common ancestor of bacteria and yeast turned into bacteria and yeast, there are a very large number of MUT-S homologs. And in yeast, they do very many different things. They're very close to each other. They're all manipulations of fixing DNA, but they're all slightly different from each other. And that makes yeast much more robust to radiation than bacteria would be. They have other ways of dealing with this. Okay, so to sort of intermediate stop here. What do we do with how do we get functional information? Well, one thing we're going to talk about DNA polymorphisms in a minute, inheritance, complex disorders, pharmacogenomics. All of these first three things we're well on the way to doing. Okay, and I'm going to tell you a little bit about that. Then the comparative genomics, orthologs and paralogs is just technical speak for what I just told you. And then patterns of gene expression, it allows us to learn a lot of stuff about what genes do. And then this other business is sort of inside baseball. Okay. So the principle we're going to use is that physically, DNA sequences have the unique property of being able to recognize each other. So that if I have this sequence over here and that sequence over there, under the right chemical conditions, only if the sequences are identical or very, very similar, they will form the famous double helix called hybridization. Okay, and if you chop up the genome of something and you do a hybridization with a probe, which is one of those sequences, and you can actually see differences. So there are enzymes that cut DNA, and this one, this is meant to be one of these is one chromosome, the other one is the other chromosome of a single individual, and they differ in that all the cut sites I've shown you here, this one is present here, but is not present there. And therefore, if I look for the length of this piece, it will be longer than here. Okay? And so this is called a restriction fragment length polymorphism. And what you can see here is that these are 9 random, I forgot how many, random Utah individuals who gave blood and got consent. And what you see is that they're all different, which is interesting. And they have two alleles, as we say. And if you follow this lady's here, and her husband over here, and you look at the children, you can see that one of the children got, the son here, got the low band and the low from each parent, but the 2 girls got the upper band from mom and the upper band from pop. Okay? And what you can say is that if this happens to be right next to a disease gene, then knowing that the older sister has the gene makes it highly probable that the second sister has it too. And that's the principle of gene mapping. And gene mapping has taken off. So today, the standard catalog lists that almost 3,000 of the 4,500 or so known inherited characteristics, we have a gene in hand. That's pretty good, because in 1980 this was thought to be impossible to do. Okay? What kinds of diseases? Well, all kinds of diseases. Diseases for which you had no realistic hope of finding out anything by doing, let's say, neurobiology or physiology or something like that. It was just too obscure, but you could by finding the gene and finding the mutation. So one of the things that we learned is that, for example, the famous Huntington's disease, of which I understand you're going to hear more later, that this turns out to be a disease of mistakes in the number of repeats, of little repeats of DNA and protein in the protein. There is no way that we would have gotten this by starting with Huntington's disease and then getting to the gene. The Lou Gehrig's disease, same kind of thing. This kind of disease, and retinoblastoma was really important because it made it clear that the relationship between inherited predispositions and loss of function at the cellular level. And of course, even this august audience occasionally watches television. And if you've watched any of these shows more than once, you will understand that all of these end up in DNA evidence. And what is the DNA evidence? The DNA evidence is just a catalog of polymorphic differences between 2 individuals, and you can distinguish any individual from anybody but his or her identical twin by this method with essentially complete certainty. This is the original marker. This is a marker that's used today in a commercial forensic laboratory. This is just a bunch of statistics saying that the FBI does millions of these determinations a day and a week, and this is just to show you, this is really fun for the old timers, that some of the really old mutants, which are the old polymorphisms, which you can see by their name, you know, D5S818, right? They're still there in CODIS. This is the famous, these markers are the markers that tell the bad guys did it, okay? Now, this kind of technology also allows you to learn things other than identity, and this was a very interesting result that was gotten by people who were studying colon cancers. And what you see is here is that we have tumors and normal. Tumor and normal, tumor and normal. Normal means the DNA was gotten from blood. Tumor means it was gotten from the tumor itself, the miscreant. And what you see is, look, there's additional bands in the tumors. So what does that mean? Well, it means that one of the phenotypes, one of the characteristics of colon cancer is that they're making mistakes in their DNA that don't happen in the blood-forming cells, in normal cells. So smart yeast geneticist said, you know, if there's a gene like that in a human, then I should be able to find one in yeast by selecting for it. So they did. They found, and then they write, and then not only did they find a gene that causes destabilization, blah, 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 but they predicted, this is only a few months later, they predicted that one of the colon cancer genes would be the homologue of this. And notice the name, MSH2. If you remember, MSH2 was one of the homologues of MUT-S. And then, indeed, at the end of the day, they found the human MSH2 ortholog is, in fact, the colon cancer predisposing gene, and 90% of all inherited cancers of this type are mistakes in one or the other of these repair genes, so that you mishandle the DNA. Okay, I'm going to end now with the sort of last deliverable, which is telling apart different subtypes of tumors. And there are technical means, which because I'm running out of time, which I'm sort of going to skip a little bit over, whereby you can get patterns of gene expression, and then you can organize the patterns of gene expression by simply asking for this gene, that gene, the other gene, the next gene, which patterns are most similar to each other. This is exactly what was done to figure out which organism was most similar to each other and produce the tree that Darwin intuited. And what you see is when you do that is that you can reconstruct reality. So this is the example. This is a nice picture that sits in the Louvre. We've made it black and white. We've randomized it as if it was a bunch of genes going across. And then we've asked the computer, run our algorithm which tries to reconstruct reality from the gene pattern, and run it on this picture. And this is the first step, that's the second step. And although you can see that it's not perfect, you get a very good idea that you're looking at this Raffaello and not something else. And in particular, even in the first case, you can see the tree here, you see, which says that some detail can be gotten out of this that you simply cannot get by some kind of global analysis. And when you do that, you discover that not only can we tell one individual from another, we can tell one tissue from another by what proteins it makes. So each of these is a different tissue, and each of these is a different tumor. And from this we learn that all the breast tumors are more similar to each other than they are to any other kind of tumor, which is an important result. And that among breast tumors there are subtypes, and you can see the subtypes yourself. I mean, you see this group, the red ones here, have a different pattern than the blue ones over here. For example, they're green for this set of genes and red for that set of genes. Okay? And you can reproduce this on a completely foreign set, and all the red ones stay together, and all the blue ones stay together, and so on. And if you look at what happened to these women, you could find that if they had a blue type, they were alive and doing pretty well years later, whereas if they had the magenta or the red type, they were not. So that this was clinically important as well as biologically significant. And we could actually get further. We could make a hypothesis. If this discovery of Pat's and my group is correct, that there are several subtypes of breast cancer, then you would expect that a gene that predisposes to breast cancer might only predispose to one type. And so we had a look at that question. And when we looked at that question, here are all the red ones, and all the red ones carried one of the genes, and it turns out the blue ones carry a different gene for predisposition, okay, among the inherited. Okay, so this is useful. Again, it's a fruit of the genome. You get better diagnosis, you get earlier detection. New therapeutic agents, of two in particular, are notable, and you have a future. That's where the pharmacogenomics is going. Okay, this is just, I threw this in just to show you that, in fact, the new treatments actually work. This is one of them, and this is another one. And there are very many cancer genes, and there are very many opportunities to do this. So KIT and ABL are the Gleevec targets, and HER2 is the Herceptin target. So the issues for the future are clear. The idea of the personal genome is much in the news. People are actually getting their genome sequenced, but nobody knows how to interpret what they learn because we don't know enough about the functions of the genes yet. We have a big problem of interpretation because the problem in interpretation is actually deep, because all these interpretations are probabilistic. The whole genetics is actually probabilistic in nature, and as a consequence of that, it's very hard for people who have no mathematics to get their minds around doing the right thing, and this is a big problem for all of us and at the elementary school level. And then also there are ethical issues. And the ethical issues have to do with what can you do about it. So for the case of Huntington's, there's not yet anything you can do about it. So do you actually want to know whether you're going to get this disease? The case of colon cancer, there is something you can do about it, and so that's potentially life-saving. And the case of HER2 amplification, it's sort of in between. For 3/4 of the women, there's nothing you can do, or not. You can't do a particular drug, Herceptin, that you can't use. But for others, for the case of Herceptin, which I happen to know well because I worked at Genentech, was involved in it, it turns out that, remember that magenta line that was very bad for women? That magenta line has been turned into something that looks like the blue line because this treatment is effective for that subset of women, but only that subset of women. So that's my story, the fruits of the genome, and I'm happy to take any questions if the boss says there's time. Okay. Yes, sir.
So, as you said right at the beginning of your lecture, the fact that different animals have roughly the same number of genes is quite a surprise.
Yeah.
And you sort of suggested that there was some insight into what that meant. Do you want to amplify a bit?
Yes. Yes. What it means is that the basic mechanisms are shared, and maybe there are 3 or 4 times as many genes in the multicellular organisms as in the simplest unicellular ones. What it mainly means is that in the complicated tissues that made us think that we need so many genes to make us, the same tricks are used over and over again in slightly different combinations. Okay, so it's a combinatorial modular construction. So if you think about a castle that you might see in a toy store made out of Legos, okay, the total number of Legos actually might be quite limited, but the number of castles that you can make might be actually quite diverse. And so there is, evolution has resulted in a highly modular structure, both at how genes are turned on and off, to all the way up to how signals are transduced and how replication is controlled, and so forth and so on. And it is those things that seem to cause cancers very frequently, failures in those regulatory pieces. But everything is turning out to be combinatorial. Yes, sir.
Could you amplify on this notion of grand unified theory in biology? I know you pose it as a bit of a...
basically, it is just a very grandiose way of saying something which is really fundamental, which is that we have hard quantitative evidence that everything on this planet that lives was ultimately derived from a single source. Okay, so we started with some primordial organism, and everything evolved from that. You could not have arranged this by a natural process. Of course, supernatural processes is above my pay grade. I don't understand that part of it. Okay, but by a natural process, you are driven to the conclusion, that everything started from the same place, and that we are all variants on the same fundamental theme. Okay, so that's the grand unification. So there's not going to be a different kind of biology in some part of the biota from another kind of biology.
So it's an analog to all four forces being from a single force in physics.
Yes, you could say that. Yes. Yes.
I got the impression that when you began your work in this field, you had no idea that today there would be these fruits. Is that right? Are you surprised at where all this has led? You're toiling in the laboratory there?
Yes and no. No, in the sense that I, being educated as a geneticist, always believed that Mendel's laws and all that sort of stuff applies as well to humans as it does to Drosophila or to yeast. Okay? But we couldn't do anything about it in the human. When the opportunity arose and we figured out a way to use the variation in human sequence to learn, for example, about inheritance, which I briefly went through, there was a sort of flash of insight once we realized, oh, you can do this. Then it very rapidly became clear that, at least in principle, you could tell one individual from another, one parent from the other, disease from non-disease, and all that sort of stuff was pretty clear. Unfortunately, it was not clear to the NIH. It was not clear to the people who give the money. It was not clear to a lot of people, but to the insiders who had been toiling in this area, it was clear, and so it no longer became a surprise. That said, I should say that what has, to me, been absolutely the biggest surprise is the same thing that surprised the engineers, which is how fast it all happened. I mean, the first useful computer, okay, was made during the Second World War. Okay, we have things that are unimaginably powerful compared to the von Neumanns who made those machines, even though they knew in principle that such a thing could be done, right? And so the rapidity with which this has all happened, the idea that actually that the armed forces, they don't need dog tags anymore. All they need to know is they need to know where in CODIS to look for a particular person, and that whole thing happened with blinding speed. I mean, it happened in a small part of my lifetime. So that's the speed has been just astonishing.